Hello, class. This is Dr. Anderson, and we're continuing our discussion of metabolism, and we're going on to the post-absorptive state. Here are our learning objectives. First of all, we want to explain how the glycogenolysis, which is the breakdown of glycogen, and gluconeogenesis, which is the making of new glucose by the liver, maintains the blood glucose level for brain function during the post-absorptive state. Secondly, I'd like students to be able to describe glucose sparing and how it supports the energy needs of the body during the post-absorptive state. Students should be able to understand and explain the relationship between increased fat breakdown, called lipolysis, and beta oxidation, and the production of ketones. Now, just as insulin was the hormone of the absorptive state, glucagon is the hormone of the post-absorptive state. Glucagon works on the liver, and I'd like students to describe the actions of glucagon on the liver and how glucagon secretion is regulated. There are some other hormones that help us in the post-absorptive state: epinephrine, growth hormone, and cortisol, and they work with glucagon to maintain blood glucose level. And then we're going to finish up with something called the leptin hypothesis, or the lipostatic hypothesis of weight regulation, regulating the size of the fat stores. So the absorptive state was the first 4 hours after a meal. So beyond 4 hours after the meal, we go into the post-absorptive state, also called the fasting state. Here, catabolism predominates. Catabolism is the type of metabolism where complex molecules are broken down. So the glycogen that was synthesized is going to be broken down into glucose. The amino acid, the proteins that were synthesized are going to be broken down into amino acids, and the triglycerides that were synthesized are going to be broken down into glycerol and fatty acid. And all these things are going to be used to support the metabolism of the body when it's been a while since the person has eaten. So we're going to break down the molecules that were stored. Most cells of the body are going to use fatty acids and ketones. Remember, during the absorptive state, they were using glucose. But now they're going to switch their metabolism to that of fatty acids and ketones. And that way, glucose can be set aside for the brain. And that's what we mean by glucose sparing. Glucose is spared for the brain. Now, the liver is breaking down glycogen to make sure that there's a blood glucose for the brain. And it's also making new glucose. And the sources for that new glucose are lactate, pyruvate, glycerol, and some amino acids. This process is called gluconeogenesis, so the creation of new glucose from non-carbohydrate sources. I want you to notice that lactate, pyruvate, glycerol, and some amino acids, they all have something in common, and that is they're all 3 carbons. And 2 3-carbon molecules can be put together to make a 6-carbon glucose. All right, so this picture is similar to the picture we looked at for the absorptive state, but we have some arrows going into different directions. All right, so it's very important for the liver to maintain the blood glucose level. It's been a while since we've eaten. So the glycogen in the liver is going to be broken down into glucose. That glucose is going to go into the blood to supply tissues like the brain. And then the brain is going to use glucose for ATP. Notice the liver is going to make glucose out of some other things. So amino acids can be deaminated, turned into a keto acid, and made into glucose. Lactic acid can be made into glucose, as well as pyruvate. Glycerol from fatty acid can be shuttled into gluconeogenesis and made into glucose. So from the glycogen and gluconeogenesis, glucose is going to enter the blood and especially to be used by the brain. So glucose is spared for the brain. Now, what are other tissues going to do? They're going to run on other substrates. So the liver is going to take fatty acids from the adipose. So adipose tissue breaks down triglycerides. That's a process called lipolysis. We get a glycerol and fatty acids. Those are sent to the liver. The liver takes the glycerol to make glucose. The fatty acids are broken down through a process called beta oxidation. Now remember, long-chain fatty acids have an even number of carbons, 12, 14, 16, 18, 20, 22, etc. And so they're broken down into groups of 2. And those groups of 2 come together to make a group of 4. That is called a ketone. And that ketone can be put in the blood. And look, other cells or other tissues can use those substances. So something like the heart can use fatty acids, lactic acid, and ketone bodies to make ATP. Other cells of the body are going to be using those ketones and fatty acids to make ATP. And they're going to be breaking down their proteins and amino acids to send back to the liver. All right, so we have glycogenolysis and gluconeogenesis and ketone synthesis going on in the liver. We have other cells running on fatty acids and ketones. We have fat undergoing lipolysis, sending their substrates to the liver for gluconeogenesis and ketone synthesis. And muscle can use its glycogen and use its own proteins to supply amino acids to be sent to the liver. Muscle can run on fatty acids, and muscle has its own glycogen. Now the thing is, when you break down glycogen, you produce something called glucose 6-phosphate. Well, you know what? The muscle cannot take off that phosphate group. Now the liver can. The liver can take glucose 6-phosphate, take off that phosphate, make glucose, and put it in the blood. But the muscle can't do that. But what the muscle can do is take that glucose 6-phosphate and put it into glycolysis and make pyruvic acid. And pyruvic acid can be sent to the liver, or pyruvic acid can be turned into lactic acid and sent to the liver. So notice we have a lot of breakdown going on. So breakdown reactions or catabolic reactions are the predominant kind of metabolism going on here. So we've looked at this picture as a whole. Let's break down each part so that you can see the words, and then we'll come back together and look at that picture again. So what kind of reactions are happening in the post-absorptive state? We have glycogenolysis, the breakdown of glycogen in the liver, glycogenolysis in the muscle, lipolysis in adipose tissue. All kinds of cells are undergoing protein catabolism. So glycerol from adipose tissue, amino acids from a protein catabolism, and other substrates, those are sent to the liver. And the liver performs something called gluconeogenesis. Gluco as in sugar, neo as in new, genesis as in creating new glucose out of non-carbohydrate sources such as lactic acid, pyruvate, some amino acids, and I neglected to put glycerol in here. Now, what do those compounds all have in common? They're 3 carbons. So the liver can take a 3-carbon lactic acid and a 3-carbon pyruvate or whatever and put those together into a 6-carbon glucose. So what happens to fatty acids? Fatty acids are broken down by beta oxidation. They're broken into groups of 2. And those 2 carbons could become acetyl-CoA and go into the Krebs cycle, or two carbons could join two carbons and form something called a ketone. An example of a ketone is, now that I just said that, acetoacetic acid, beta hydroxybutyric acid. Acetone is an example of a ketone. So these are fatty molecules that actually many cells of the body don't have any problem running on. But the thing is, they're in groups of two. And 2 and 2 don't make 6. So fatty acids cannot be used to make glucose. Fatty acids can be used to make ketones, and ketones can be used to make ATP. But when cells are burning fatty acids, they cannot make new glucose out of that. So another way to say that is fatty acids are not gluconeogenic. All right, role of the liver. Glycogen is broken down into glucose. Glucose is able to exit the liver via a GLUT transporter. Now the liver's kind of interesting because when insulin is gone, the GLUT transporter doesn't go away because it's a different variety in muscle and some of those other tissues. It stays there, but it allows glucose to go in an opposite direction. So in the absorptive state, glucose is coming in via the GLUT transporter and being stored as glycogen. During the post-absorptive state, we're breaking down the glycogen and glucose is exiting the liver. That means we have glucose for the brain. Now the liver makes 3 carbon molecules such as pyruvate, lactic acid, glycerol, some amino acids, and makes a 6 carbon glucose. That's gluconeogenesis. Fatty acids are broken down via beta oxidation. That forms ketones, and then ketones are a major source of the body. So fatty acids are ketogenic. Pyruvate, lactic acid, glycerol are gluconeogenic. You cannot make glucose out of fatty acids. So glucose sparing. During the post-absorptive state, most cells of the body convert to fat metabolism, meaning they're using fatty acids and ketones. As the result, the rest of the body isn't hogging all the glucose. Glucose can be saved for the brain, and that process is called glucose sparing. So many parts of the brain, particularly the higher centers, they need glucose. Neurotissue needs glucose. Now, some parts of the brain can use ketones. So they can use ketones and glucose. But the brain really prefers glucose. And I need you to understand that the brain can't use fat. The brain, the neurons are just not geared for fat metabolism. And the thing is, the brain uses lipids for other things. The brain uses lipids to make structures. For example, myelination. Myelination is a huge use of lipids. And then the liver makes, the brain makes all kind of neurolipids and sphingolipids and all that sort of thing. So the lipids in the brain are part of how the brain signals and all that sort of thing. So the brain cannot be digesting itself. So the brain does not run on fat. It prefers to run on glucose. It can run on keto acid or ketones if you make it, but glucose is the preferred substrate. The rest of the body then is going to run on fat because the rest of the body has no trouble running on fat. Now the muscle. The thing is, fat that we break down doesn't have enough glycerol to supply the liver with the 3 carbon molecules it needs to make new glucose. There just isn't enough fat for that. So the muscle breaks down some protein, and those amino acids are sent to the liver for gluconeogenesis. Muscle has glycogen. Glycogen is broken down into glucose 6-phosphate, can be put into glycolysis, and that could be fully burned in the muscle, or the products of glycolysis, pyruvate or lactate, can be sent to the liver for gluconeogenesis. Triglycerides are being broken down during the post-absorptive state. They're broken down into a 3-carbon glycerol backbone and 3 fatty acids. Glycerol is sent to the liver for gluconeogenesis. Fatty acid is sent to the liver for beta oxidation and ketone synthesis. And then fatty acid is sent to other tissues for ATP synthesis. How does the body know to be doing this? Well, we have a hormone produced by the alpha cells in the islets in the pancreas, and that hormone is called glucagon. And when plasma glucose goes down to low levels, we get a negative feedback control of blood glucose level, because that's the signal to release glucagon from the alpha cells. Plasma glucagon goes up in the blood. Now, glucagon works on the liver. Works on the liver, works on the liver. Did I mention that glucagon works on the liver? Yes, it works on the liver. And it's going to do 3 things. We're going to break down glycogen, so there's new glucose for the blood, perform gluconeogenesis, so there's new glucose for the brain, and we're going to produce ketones, so the rest of the body has something to run on. So as a result of glucagon, and this is a picture from a textbook called Vander. So we're going to have an increase in glucose and an increase in plasma ketones. So notice negative feedback. Plasma glucose is low. Glucagon works on the liver to get plasma glucose back up again. So glucagon has functions that are antagonistic to those of insulin. Insulin helps make glycogen. Glucagon helps break it down. Insulin prevents gluconeogenesis. Glucagon promotes it. Insulin causes synthesis of triglycerides. Glucagon causes its breakdown. Insulin promotes protein synthesis. Glucagon inhibits it. So glucagon binds to its glucagon receptor in the liver. Here's a hepatocyte. Notice this is a G protein coupled receptor. We get cyclic AMP from adenylyl cyclase. Cyclic AMP then activates protein kinase A, and we promote the enzymes that are going to make glycogen. So we phosphorylate the glucose and hook them together. And glycogen is shown as kind of a okay, I'm suddenly worried that I had a brain fart and I just misspoke. And this is a presentation that I cannot go back and edit. So I'm just going to retrace my steps to make sure that I didn't misspeak. So glucagon binds to its receptor, activates a G protein, which activates adenylyl cyclase. We activate cyclic AMP. Cyclic AMP promotes the action of a kinase. Kinases are enzymes that phosphorylate things. So here's glycogen phosphorylase, and it gets phosphorylated. And now that I am going through this, I think I did misspeak. And so then we break down the glycogen. We break down the glycogen, remove the phosphate, and we've got a little glucose. And look here, our gluc transporter is there in the membrane, but instead of having glucose come in, glucose exits and provides glucose for the brain. So insulin and glucagon are antagonizing one another in terms of blood glucose level. So remember, during the absorptive state, we had hyperglycemia. That stimulates the beta cells. Then we get insulin. Insulin promotes the use of glucose and the storage of glucose. Then blood glucose level goes back down to where it should be. If blood glucose level gets too low, then we're going to inhibit the release of insulin. And if blood glucose level goes down even more, then we're going to stimulate glucagon. Glucagon is then going to work on the liver to break down glycogen and to produce new glucose. The blood glucose level goes up. If it continues to go up, then we'll inhibit glucagon. And then if the blood glucose gets too high, then we're back to insulin again. So hopefully that you'll see this is a negative feedback mechanism centered around regulating blood glucose level. So post-absorptive state regulation, you see breakdown of glycogen in hepatocytes and skeletal muscle brought around by glucagon. Epinephrine also does this as well. Lipolysis in adipocytes is broken down by epinephrine, norepinephrine, and then we have other substances, cortisol, insulin-like growth factors. Some of these are permissive effects, and likewise. Notice what you don't see here, glucagon. Glucagon does not work on adipocytes. It only works on the liver. Protein breakdown, most cells of the body, that's a cortisol effect, and gluconeogenesis in hepatocytes. It's not, there is some gluconeogenesis that happens in the kidney, but in this course, we're not going to get all involved in this. In this course, we're going to center our conversation around the hepatocytes. And glucagon with the permissive effects of cortisol are going to promote gluconeogenesis and the increase in blood glucose levels. So notice we have some permissive effects. Growth hormone interacting with epinephrine, growth hormone being permissive for glucagon, cortisol being permissive for glucagon, and those sorts of things. But notice all the things that are happening to raise blood glucose level because you haven't eaten for a while and your blood glucose level is low. All right, the last thing I want to talk about is a hormone called leptin. Leptin is a hormone that comes from your adipose tissue. So it turns out your adipose tissue is an endocrine gland. Who knew? So the more fat stores you have, the more you make leptin. The smaller your fat stores, the less you make leptin. And so it's been proposed that there's a negative feedback mechanism that sort of controls somebody's fat stores. So you eat some food, you make some fat, your fat secretes leptin, leptin goes in the blood and works on your hypothalamus. And what it does is it lowers your food intake and increases your metabolism. So if you gain some weight, then you are decreasing your food intake, increasing your metabolism. Then if you lower your food intake and increase your metabolism, then you lose your fat and get your leptin back to where it should be. So people that lose a little adipose tissue, they'll lose leptin, their appetite goes up, their metabolism goes down, and they gain the weight back. Or they gain some weight, they produce more leptin, their metabolism goes down, or their metabolism goes up, their food intake goes down, and they lose the weight and they get back. So it's sort of the lipostatic hypothesis of weight control. So an interesting thing. Some mice were discovered that are genetically deficient in leptin, and they're obese. I mean, they are big, fluffy mice. And if they are given leptin, they lose weight and they become little skinny mice. Well, the scientists who discovered this said, well, we're going to make $1 million and help people lose weight. So they did some clinical trials. There were people with obesity and they gave them leptin. Now, they lost a little bit of weight, but not very much, like 5 pounds or something, not very much weight at all. And then when these scientists checked people who struggle with obesity, turns out they have plenty of leptin. They actually have high leptin in their blood. So what's going on? The mice didn't have leptin, but the people do have leptin. So why aren't the people's hunger going down and their metabolism not going up? So I'm going to just give you a minute to think about it. Did anybody come up with the idea that these folks have leptin, but they're not responding to it very well? So there's a downregulation in leptin receptors, and there's less of a response to the leptin in the blood. So we got to figure out a way. One way to help people that are obese might be to figure out a way to help their bodies be more responsive to leptin. And so it is really, really hard to lose weight. I struggle with losing weight. And the thing is, you lose a few pounds and then you're just ravenously hungry. And why is that? You've lost a little bit of weight, your leptin has gone down, and your appetite goes up. And so then it's hard to maintain the weight loss. Trying to lose weight, certainly you have to eat less and exercise more. However, that is easier said than done. And so here's one little picture about the complexity of metabolism and weight loss and weight control. Alrighty, so I made this presentation in the spring of 2017. So this particular day we're doing evaluations, and I hope that you turn those in. And Friday coming up, we're going to do temperature regulation. So once again, I appreciate very much that you're taking the time to listen to this presentation and to dig into your physiology learning objectives. Please let me know how I can help. If you have a question, by all means, put them on the forum, and I'm happy to help. Keep doing your critical thinking, keep retrieving, keep working with your study buddies, and I'll see you next time in class.